Do you know who the protists are? If not, don't worry. No one really knows for sure. Scientists have only loosely agreed that if an organism is made of a single cell and has a nucleus, we can call it a protist. And that's where all the chaos comes from. Some forms stretch out like green little spindles and carry chloroplasts. Some are fluid and ever-changing like a living drop, and some are airy, glass spheres covered with the finest needles, along with many other equally astonishing beings. So how do we bring order to a kingdom where we ourselves have created such a fantastic mess? In all this chaos, it's easy to get lost scientists argue endlessly about which group each organism belongs to. So let's take a step back and make things simpler. We'll group the protists. Let's choose the 6 most recognizable ones: flagellates, amoeboids, heliozoans, radiolarians, ciliates, and foraminiferans. But how can we tell them apart under the microscope? The first thing that comes to mind is shape. It seems like that should help, but it's not that simple. See for yourself how easily they can be confused. Here are the amoebozoa. They are the only ones that can be distinguished really easily. Only they have thick, broad lobopodia. And here are the flagellates. But flagella are not exclusive to them. Some amoeboids have them too. Maybe the shells. But they are found not only in foraminiferans, but also in some amoebozoans. The difference is that in foraminiferans, the shell is covered by a thin layer of cytoplasm, while amoebozoans do not have this feature. Under a light microscope, you won't see this difference anyway, so the trait is not a reliable one. A radiating body shape? That's not just the heliozoans, but also the radiolarians, and even the red amoeba, Vampirella. Attached forms? These can include both the trumpet-shaped ciliate and the heliozoan Clathrulina. And cysts are the greatest mystery of all. Tiny, round, and almost identical. For example, here's a cyst of an entamoeba and another one from a ciliate. A single external feature rarely allows you to determine the group with certainty, but the overall appearance of the cell and a combination of simple morphological details provide very reliable clues. Let's begin with the amoebozoa. Amoebozoans are characterized by their thick, broad lobopodia. This is the first feature by which they are usually recognized. The large, pale, rounded spot inside the cell is not the nucleus, but the contractile vacuole. It periodically fills with water and then contracts, helping the amoeba remove excess fluid. The nucleus is present as well, but it looks different. Usually smaller, darker, and more oval, and on fresh slides, it is not always easy to see under the microscope, especially in fast-moving forms. Some species have several nuclei. In the cytoplasm, you can see digestive vacuoles. If the amoeba has recently fed, there may be many of them, from 1 or 2 to a whole scatter. The greenish tones come from the digestion of captured algae. Also, pay attention to how cytoplasmic density is distributed. Near the edge, the cell looks lighter and more transparent. This is the ectoplasm, a thin outer layer that helps form the pseudopodia. Deeper inside lies the endoplasm, more granular and fluid, containing most of the organelles. Now let's look at the foraminiferans. All foraminiferans have a shell covered on the outside by a thin layer of cytoplasm. Because of this, their skeleton is essentially internal rather than external. The cytoplasm of foraminiferans can extend outward through many small openings on the surface of the shell rather than through a single opening as in testite amoeba. The shell of foraminiferans is almost always multicameral. New chambers are added one after another as the organism grows, and small openings remain between them. Through these openings, the cytoplasm flows from one chamber to the next. These openings are called foramina, from the Latin foramen, opening. And this is why the group is named foraminifera, meaning bearing openings. The smallest central chamber is the first one, and all subsequent chambers gradually grow around it. Foraminiferans also have pseudopodia. These are called reticulopodia. They are extremely thin, branching cytoplasmic threads that can merge into a complex network surrounding the shell. Small granules constantly move along these threads, making the living specimen appear as if particles are streaming across the network. The shells of foraminifera preserve exceptionally well, and entire rock formations are made from their fossilized remains. The most impressive are the giant nummulites, ancient foraminifera the size of coins, which were even used as building material in the pyramids of Giza. This is not a sunrise. This is a heliozoan. Heliozoans are easy to recognize by their characteristic radiating projections, the axopodia. These are thin, rigid rays containing straight bundles of microtubules that converge at the center of the cell. The cytoplasm of a heliozoan is divided into two layers. The ectoplasm is the lighter outer layer that contains the bases of the axopodia. This is also where the contractile vacuoles are located. As in other protists, they help expel excess water from the cell. Inside lies the endoplasm, denser and more granular. Here you find the nuclei, and there are many of them. Small digestive vacuoles move toward the central part of the cell. This is a radiolarian. Does its shape seem simple to you? In acantharians, the shape truly is simple. Their strontium skeleton consists of just a few rays spreading out like a star. But in other radiolarians, everything is much more interesting. Their silica skeletons resemble miniature spaceships or intricate Chinese puzzle balls. Heliozoans also have siliceous spicules, but these never form a unified lattice. The axopodia of both radiolarians and heliozoans contain microtubules, but in many radiolarians, axopodia run inside the rigid channels of their silica skeleton. Because of this, the external rays appear thicker. The skeletons of radiolarians preserve beautifully in the fossil record. They are found starting from the Cambrian. Radiolarians have another distinctive feature. Their cell is divided into two parts by a central capsule, a dense organic shell with pores. Inside the capsule lies the denser endoplasm. The endoplasm contains numerous tiny nuclei in addition to organelles. Outside is the ectoplasm filled with bubbles and vacuoles. The skeletons of acantharians are made of strontium sulfate, and they dissolve in seawater, almost without a trace. In contrast, the silica skeletons of radiolarians dissolve much more slowly. After the cell dies, the empty skeletons sink and accumulate, forming enormous deposits of radiolarian ooze. Over time, these sediments can transform into siliceous rocks such as opal, chalcedony, and microcrystalline quartz. Ciliates are one of the easiest protist groups to recognize. First, their entire body is covered with cilia. They are small, but still visible under a light microscope. The second feature that stands out is the presence of two contractile vacuoles. In almost all ciliates, the vacuoles are arranged symmetrically and work in opposite phases, contracting and expelling excess water. In a healthy ciliate, if one vacuole is small, the other will be enlarged. The third hallmark is the presence of two nuclei. The large nucleus is called the macronucleus and controls the cell's day-to-day functions. The small one, the micronucleus, takes part in the exchange of genetic material between ciliates. Many ciliates also have an interesting structure called the cell mouth, or cytostome. This is an invagination through which food particles are guided into the cell and packaged into digestive vacuoles. The shape of the cytostome varies widely, from a simple funnel to a complex spiral structure. And finally, the flagellates. Euglena is not the most typical representative of this group. It has chloroplasts, so it is capable of photosynthesis just like plants, and a small red eye spot helps it swim toward sources of light. But the main feature that defines the flagellates is the flagellum. Euglena has one. There are actually two, but the second one is rudimentary and not visible. Under a school microscope, a flagellum appears as a barely visible, transparent thin filament. Most often it is easier to detect it by the characteristic movement of the cell than to observe it directly. Nevertheless, the flagellum remains the main feature used to identify the group of flagellates. You might exclaim, "But amoebas have flagella too." And you would be right. That's why, to reliably distinguish different groups, it's important to look at how the cell moves. Even if an amoeba has a flagellum, it still moves in an amoeba-like way, using broad, flat lobopodia, which it also uses to capture food. Interestingly, amoebas move thanks to the proteins actin and myosin, the same ones that work in our muscles. At the front edge of the cell, actin quickly assembles into a dense gel, and a pseudopodium pushes forward. Behind it, the actin network disassembles, the gel turns into a more fluid sol, and the cytoplasm flows toward the growing region. As a result, an amoeba doesn't move simply by oozing around like snot, but thanks to alternating zones of actin assembly and disassembly that constantly shift the leading edge of the cell. If you look at an amoeba from the side, you can see that it doesn't just spread across the surface, but almost walks on its pseudopodia, slightly lifting itself above the substrate. Foraminifera also form pseudopodia, but they look nothing like those of amoebas. Their thin strands are called reticulopodia, and they create a real mesh-like web around the shell. Tiny granules constantly move along these strands, in both directions. It looks like little wagons moving along rails. This bidirectional flow is driven by actin dynamics and cytoplasmic streaming inside the thin extensions. Such a network allows a foraminifer to cover a large area around itself, to catch food particles, transport them to the shell opening, and even pull itself forward slightly by using different parts of the network. Heliozoans can move using their axopodia, thin, straight rays that are much stiffer than the reticulopodia of foraminiferans. Inside each axopodium runs a strong bundle of microtubules, and it is this structure that gives the ray its rigidity. The microtubules can slowly assemble and disassemble, and motor proteins sliding along their surface can slightly shorten or bend the ray. Thanks to this, an axopodium can retract into the cell if a bacterium or another particle sticks to its tip. From the outside, it looks as though the prey is riding along the axopodium, like on an escalator, straight into the heliozoan. Heliozoans move extremely slowly. They can walk along the substrate by retracting some axopodia and extending others, or redistribute their cytoplasm so that one side becomes heavier. And then the whole cell seems to roll a few microns in that direction. A heliozoan is ready to eat an alga, a ciliate, fungal spores, or even a small multicellular animal. On the surface of its axopodia, lie tiny sacs filled with sticky material, extrasomes. And then everything unfolds like a scene from a horror movie. The unfortunate prey accidentally touches a ray. The nearest extrusomes instantly discharge their glue onto it, and the ray to which the victim is stuck begins to contract, pulling the prey toward the predator's body. Heliozoans lack a cellular mouth. Whenever a prey particle touches the cell surface, it's engulfed exactly at that point and digested alive. The more selective species deploy toxic extrasomes, killing their prey instantly. Large prey doesn't intimidate them. Groups gather around, encircle it, and digest it cooperatively inside a massive shared food vacuole. Radiolarians are able to move, but very slowly. Inside each axopodium, microtubules undergo continuous local assembly and disassembly, while motor proteins migrate along their surface. This allows the ray to make tiny structural adjustments in length or angle without losing its mechanical stiffness. By these microshifts, a cell can pull attached food particles closer or gently reposition itself by a few microns. Some species also capture prey using adhesive or toxic extrusomes. Flagellates are not just able to swim. They can maintain a stable height in the water, turn in place, abruptly change their trajectory, and respond to light, chemical cues, and obstacles. Some flagellates have two flagella. One pulls the cell forward while the other acts as a rudder, providing stability and allowing the cell to turn instantly. Although both eukaryotes and prokaryotes have flagella, their structures are completely different. A bacterial flagellum is a rigid filament made of the protein flagellin. It rotates like a propeller around its axis, pushing the cell forward. Its motor is built directly into the cell envelope and runs on an ion flow. A eukaryotic flagellum is built in a different way. Inside it lies the axoneme, 9 doublets of microtubules surrounding a central pair. This is the famous 9+2 pattern. Movement depends on ATP. Dyne motors use its energy to make the microtubules slide against each other, and the flagellum bends, producing soft, wave-like, whip-like motion. Ciliated protozoans move using thousands of short cilia that cover the entire surface of the cell. Each cilium is built in the same way as a eukaryotic flagellum. But the most interesting part is that their movements are not chaotic. They follow strict coordination rules. If you look at a single cilium, you can see that it moves asymmetrically. First comes a powerful, effective stroke that pushes the cell forward, and then the cilium smoothly returns closer to the surface during the recovery stroke. Beneath the membrane, the cilia are linked by rootlet structures called kinetodesmata. They connect the cilia into long longitudinal rows and help transmit movement from one cilium to the next. As a result, metachronal waves seem to run along the surface of the cell. Together, this creates smooth, rapid, and highly controlled movement. A ciliate can not only swim straight, but also change speed, turn precisely, and even collect food by directing water currents toward its oral region. Reactions to stimuli in ciliates are astonishingly fast, even though they have no nervous system. Any mechanical or chemical signal instantly changes the beating pattern of their cilia, allowing the cell to orient itself in space and maneuver efficiently. Turns are created by adjusting the angle of ciliary beating along the sides or at the front of the cell, as if the ciliate is steering itself with tiny water currents. Scientists at Stanford University even managed to turn a ciliate into a character in the video game Pac-Man. They applied very weak electrical impulses to a chamber with paramecia. If a ciliate swam in the wrong direction, the impulse nudged it to turn back. As a result, a person could literally control a ciliate from a keyboard, guiding it around obstacles and along a chosen path, like a tiny living game character. We've already explored all the key characteristics of protists, and the last thing left to discuss is how they reproduce. Most protists do it quickly and simply by dividing the cell in two. Amoebas reproduce this way, for example. If an amoeba has a shell, it pushes a large bud out through the aperture, forms a new shell on it, and afterwards each amoeba begins its strong and independent life. Heliozoans, radiolarians, flagellates, and ciliates also reproduce by binary fission. Some heliozoans and radiolarians can also reproduce by budding, when a small daughter cell detaches from the parent. Ciliates have an additional process called conjugation, which does not increase the number of cells, but serves as a way to exchange genetic information. Two ciliates come together and attach at their cytostomes. The macronucleus breaks down, and the micronucleus undergoes meiosis. 3 of the 4 resulting nuclei disintegrate, and the remaining one divides once more by mitosis. One of these nuclei crosses the bridge into the partner cell and fuses with its nucleus. A new macronucleus then forms, and the partners separate. The most complex case is found in Foraminifera. They have an exceptionally intricate life cycle with alternating diploid and haploid generations. But that, as they say, is a story for another time.